morning and welcome to a special episode of Science Radio. We present to you a series of 3 episodes where we will cover a workshop on Hippocampus titled From Synapses to Behaviour, held at IISER Pune from the 1st to the 3rd of December 2013. We will hear from some of the leading neurobiologists around the globe about the lectures they delivered at the workshop. Find out what motivated them to take up neuroscience and what they feel about future prospects of research in neuroscience in the country. Apart from all the serious science talk, the workshop hosted a musical night featuring the genius of percussionist Ustad Tawfiq Qureshi. We were fortunate to have had the opportunity to chat with him. We will hear from Ustad Tawfiq Qureshi about the wonders of music, his love for food, and a lot more. This is Science Radio on Vidyavani 107.4. First, we will hear from Dr Collins Icci, Assistant Professor of Biology at the Indian Institute of Science Education and Research, Pune, and Dr Suhita Nadkarni, who is a Wellcome Trust DBT Fellow. Dr Icci and Dr Nadkarni were the local coordinators of the workshop on Hippocampus, From Synapses to Behaviour. We caught up with them in the studio at IISER Pune. We are in conversation with Dr. Collins Icci and Dr. Suhita Nadkarni. Welcome.
Thanks.
The workshop was organized by Indian National Node for Neuroinformatics. Could you please tell us a little about it?
The Indian National Node for Neuroinformatics is an organization that sort of attempts to bring together scientists working in the field of neuroinformatics, which is basically an amalgamation in some sense of neuroscience and information technology. The Indian Node was sort of initiated in an attempt to provide a forum for researchers to get together and talk to their peers about neuroinformatics in general, also to share the tools that are constructed. It has an educational component to it in the sense there are workshops in schools that are organized. Essentially, it's a place where this workshop is an opportunity for us to invite a lot of the leading minds in the field to come and talk and interact with the scientists and the students at our institute and talk about the problems that interest them.
Could you tell us a little about the workshop itself? It was titled Hippocampus From Synapses to Behaviour.
In organizing this workshop, some of us had a vested interest too because we worked on Hippocampus. It's actually a useful system to look at when you address problems that define the way neuroscience has evolved over the history of neuroscience itself. Some of the problems that we're really interested in are trying to understand how the external world is represented in our brain, how we behave in this external environment, how we navigate through space, how we retain a memory of the things that pass us by, the places we visit, and so on. And the hippocampus has been a model system to look at all of these diverse problems at many different scales of organization, ranging from that of behavior down to the subcellular and the synaptic scale. In studies of the hippocampus, there has been a lot of back and forth between theory and an experiment. A lot of what we know about how single synapses work, how memories are formed, the mathematical models underlying it, our idea of representing the external world, of walking through space and how that's represented in our heads. Both experiments and models have sort of come out of studies from the hippocampus, and there's been a lot of back and forth between theory and experiment. One of the things we thought the workshop itself would be a place where theorists and experimentalists could come and talk about their work.
To add a little bit to that, even though it seems like a super specialized area, like the left ear or the right ear, but it turns out it's not. Lessons learned from hippocampus have been applied to various other systems. So it's not necessarily as specialized as it sounds.
How would you describe the overall atmosphere of the workshop?
Electric. I think it was remarkable. I haven't been to all the other INI workshops before this, but from most accounts, this was probably the most well-attended INI workshop that they've had. The number of registrants for this workshop were approximately 150. And you have to realize that the workshop was called Hippocampus, right? So to have 150 people show up to listen to talks from across the world on the hippocampus was actually quite remarkable. I think the scientific tone of the workshop was set by a number of remarkable talks all through the workshop. It was really important that students at IISER, students from all over the place, they got to speak to some of the leading lights in the field and actually discuss science and sort of get into the nitty-gritty and the workings of science, which is really very important.
Also, I think the guidelines of the conference about a substantial part of the time being spent on discussions, 30% of the time was spent on discussions. The talks were meant to be somewhat pedagogical, and that helped, I think, keep the general interest of the audience.
Thank you very much for giving us your time. Prashant Biraka and Sadhna Panzare are here with me today to take you through the workshop on Hippocampus, from Synapses to Behaviour.
Hi, this is Sadhna with Science Radio.
Hi, this is Prashant with Science Radio. The workshop saw some of the leading researchers from all around the world come together and share their research. Among them was Dr Pender Paula from the National Centre for Biological Sciences in Bangalore. Dr. Paula specializes in emergence of memory from multi-level neuronal computation. He had some fascinating thoughts to share about the hippocampus.
Hippocampus has been a hot topic for a very long time. Hippocampus is deeply implicated in learning and memory. And there's many experiments that have shown this, ranging from behavior to physiology, but perhaps the most striking experiments were done on a patient by the name of H.M. Henry Molaison. It's turned out in the past few years, since he died, they could release his name. He was the unfortunate victim of a form of epilepsy, which at the time the doctors felt that the best way to cure was to do surgery, rather dramatic surgery, in which they removed his hippocampus. And the outcome of that was quite remarkable. It was that he was unable to form new memories. He was perfectly capable of remembering things that had happened to him well before his hippocampus was removed, but he was completely unable to remember new memories, so much so that, in fact, the scientists and doctors who studied him in the subsequent decades had to introduce themselves every morning. He just remembered nothing at all. By all accounts, he was a very good-humored and gentle man, but of course he was adrift in the world because he had no memory of it for the decades that he lived after his surgery. So that is an example of what happens if you don't have a hippocampus. And since then, along the years, many experiments, what happens to the learning of mice and rats if their hippocampus is damaged, what happens to if there are drugs given that block the activity of certain cells in the hippocampus. All of these things strongly support the idea of its role in memory.
The title of Dr. Bhalla's talk was Watching Memories Form by Imaging Hippocampal Activity During Learning. Let us hear what he had to say.
This was work that was done by my student Mehrab Modi, based on the development of a very nice 2-photon in vivo imaging system by another student, Ashish Dhavale. So what the experiment was about was the following. We trained a mouse, or a series of mice, to associate a sound with a subsequent puff of air to the eye. And of course, when there's a puff of air to your eye, you blink. And this is very similar to Pavlovian conditioning. And so the animal learns that if the sound comes, it should blink. The key thing was that we did this training while its brain was being imaged in a manner which let us watch the activity of cells in the hippocampus and watch the activity of hundreds of cells in the hippocampus at the same time. So the question then is, what changes in this activity when the animal learns to blink when the sound comes, even before the puff of air? And so, well, what we find was that there's a sequence of activity of cells that the animal starts to generate as it learns, and we also find that there's patterns of correlated activity which relate to how the connections in the hippocampus are set up, and those change during learning.
This is Science Radio on Vidyavani 107.4.
Dr. Trygve Solstad from Kavli Institute for Systems Neuroscience in the Norwegian University of Science and Technology specializes in neurophysiology and mathematical modeling of neural networks. He presented a captivating lecture on neural code for spatial localization. Let's find out more.
I'm working on a brain area called the hippocampus and a neighboring area called the entorhinal cortex. And these areas are very important for our memory and for how we find our way in the real world. And what's special about them is that they have some neurons that encode maps of space and form maps of our memories. So we can record the activity from single cells in these areas in a rat that runs around in a maze. And listening to only a single neuron runs around in this maze, a single neuron is responsible for telling the rat where it is in a particular area in this maze. So when we look at several neurons, we can tell where the rat is only looking at this neural activity. So together they form a spatial map of this box. When we put the rat back in the box a few days later, the same neurons will be active in the same positions in this box, which tells us that these neurons remember the map of this box over several days. So it's a long-term memory of this maze. Now there are several cell types in this brain areas that work together, and they all have different tasks. So this one neuron I told you about, we call them place cells because they have a particular place in the box. Then there are cells that we call head direction cells, and they are more like a neural compass. So each cell has the task of telling the rat when its head is pointing in a specific direction. And so you have a third type of cell that's called border cell, which we also discovered in the Kavli Institute, that has the job of signaling when the rat is running along borders of the environment. For instance, a wall, or if there's a drop, or it's something the rat cannot pass. So what we're doing in the Institute is trying to map out what kind of cell types exist in these brain areas. So that's sort of the building blocks of memory, you could say. And then we would like to know how they work together to form maps and how they are used by the rat to navigate and solve different tasks.
From the Indian Institute of Technology, Kanpur, Dr. Janaki Sen, whose primary research interest lies in understanding pattern formation in the developing nervous system, presented her talk on insights gained from chick embryo about the development of hippocampus. Let us hear from Dr. Sen.
It's about looking at the hippocampus and how it develops, and we are using the chicken embryo as a model system to do it. And I think our group may be the only one who's doing that. Very few people work on the chicken embryo in the country. I don't think anybody else works on neural development, let alone the hippocampus, with the chicken model. We have established the chicken, we have characterized its development essentially at the molecular level, and now we have a handle on how to study the changes that are happening throughout development, and we want to pursue that further.
The workshop also featured Dr. Tom Bartol from Salk Institute in California. Dr. Bartol specializes in developing computational models on biophysical models of synapses, dynamics of processing in neural circuits, and learning and memory in neural systems. His talk was on how to build a synapse from molecules, membranes, and Monte Carlo methods. Here is a short summary from Dr. Bartol.
What I presented today was our work in putting together a very realistic model of all the molecules that we think are involved in synaptic transmission, from the presynaptic release of neurotransmitter to the postsynaptic activation of receptors and what happens downstream from there. The complexity of these systems is such that it's really hard to think about how all these different molecules that other neuroscientists have been discovering that are involved in this process, how they all work together to produce the integrated function of the whole system. And I made the point in my talk that without a good intuition about how these things interact, it's hard to think of a hypothesis about what experiment you might do, how it might work, and what experiment you might do in order to see whether you are right about those things. And without intuition, you can't do a thought experiment and make a hypothesis. So computational modeling is a way to put the pieces together in the way that you think they're put together from existing studies, and then see whether they really will produce a result that replicates an experiment. And through this process, you gain intuition about the system. And from that intuition, from the model, you can then go back to experiment. So the model is not supposed to take the place of reality. It's supposed to be a playground in which you try out different things, and the purpose is to gain intuition and understanding of the system.
Dr. Mayank Mehta is a researcher from the University of California, Los Angeles, and works on computational and systems neuroscience, learning and memory, sleep, neural oscillations, and neural coding. Dr. Mehta delivered a wonderful talk on multisensory control of hippocampal activity and multimodal behavior. This is what Dr. Mehta had to say.
The basic question that we were interested in is how is it that we form maps of space and how do we keep track of time? And this is something all animals do. They have to navigate in the world because they're animate, they have to walk. What is not known is what exactly makes up space and how the brain makes up that space, and can we figure this out? So when the animals walk around in the real world, all kinds of things change. As soon as the animal takes a step, the sound changes, the light changes, the smell changes, the texture changes, and it's kind of uncontrolled. So we thought in order to really understand this, we need to now do a multimodal virtual reality where the rat is on a ball that's floating and he takes a step, but the world, the sound and the light and the smells that change, they are under our control. So using a computer, we take every movement the rat makes and then project an image and sound and smells based on what we want the world to be. So now we have control over those stimuli, and we can take away one or the other or put all of them in, and we can now understand how are they perceiving space.
Finally, we spoke to Dr. Vidita Idea from Tata Institute of Fundamental Research at Mumbai. Dr. Vaidya works on the neurobiology of emotions and had some interesting facts to share.
We know a lot about the building blocks of the brain. We know about how neurons get to the locations that they're supposed to, how they connect up and form circuits. We understand a fair bit about how we perceive sensation, how we control motion, how we may actually move our hands and our legs. Even autonomic functions like breath control, we have some understanding of. But relatively, we still have a poor understanding of the circuits in the brain that regulate the things that we really identify with our own individual identity, with our feelings about ourselves, our environment, our peers, our loved ones. We really don't have a good understanding of the circuits that mediate that. We have, to some extent, a better understanding about negative emotions. So we have a reasonable understanding of the circuits that modulate relatively evolutionarily conserved emotions like fear. But we don't have a good understanding of circuits that regulate joy and emotions like happiness. Certainly laughter and humor, the circuits that mediate these, are poorly understood. Also, circuits that regulate things like attachment and affection are again not really well understood. And I find this fascinating because I think that neuroscience has now matured enough to allow us to now move into understanding these circuits that mediate social behaviors, which form a large part of who we are. So yeah, that's what I do. I study rats and mice, and I try to understand circuits that may be modulating, in particular, attachment behaviors and the early patterning of how the quality of attachment may change long-term behavior.
This is Prashant.
And this is Sadhna.
Signing off.
That's all the time we have for this week's episode. Tune in again next week for another episode from the series Hippocampus, from Synapses to Behaviour, where we find out how these researchers got interested in neuroscience and what the future prospects of neuroscience research are in India. Also, a look at the poster presentation held as a part of the workshop. This is your host, Saurajit Basu, signing off.